Functions. Mitochondria are main sites of aerobic respiration. They are miniature biochemical factories where foodstuffs or respiratory substrates are completely oxidized to form carbon dioxide and water. The energy liberated in the process is stored in the form of adenosine triphosphate. These bring about the oxidation of carbohydrates, proteins, and beta oxidation of fats. The energy ATP produced in the mitochondria helps to perform various energy requiring processes of the cell like muscle contraction, nerve impulse construction, etc. Because of the formation of adenosine triphosphate, mitochondria is called as the powerhouse of the cell. Plastids. Plastids are found in all plant cells and the euglenoids, etc. These are easily observed under the microscope as they are large, depending upon their color and their pigments they contain. These are of three main types: leucoplasts, chromoplasts, and chloroplasts. Leucoplasts. These are colorless plastids which generally occur near the nucleus in non-green cells. They have variable size, form, and stored nutrients. Granum is absent. There are three types of leucoplasts: amyloplasts, elyloplasts, aleuroplasts. They are amyloplasts. These are starch-containing leucoplasts. Example: potato, tuber, rice, etc. Elyloplasts. They store fats and oils. Example: castor. Aleuroplasts. These leucoplasts store proteins. Example: aleuron cells of maize. Chromoplasts. These plastids are yellow, orange, or reddish in color because of the presence of carotenoid pigments. Chromoplasts are formed either from leucoplasts or chloroplasts. Change of color from green to reddish during ripening of tomato and chili is due to transformation of chloroplasts to chromoplasts. The orange color of carrot roots is due to chromoplasts. Chloroplasts. They are greenish plastids which possess photosynthetic pigments, chlorophylls and carotenoids, and take part in synthesis of food. Majority of chloroplasts of the green plants are found in mesophyll cells of the leaves. They vary in size, shape, and number. Size. They have variable thickness of 2 to 4 micrometer and length of 5 to 10 micrometer. Shape. They may be spherical, lens-shaped, oval, discoid, or even ribbon-shaped in some plants. Number. Their number varies from 1 per cell in Chlamydomonas, a green algae, to 20 to 40 per cell in the mesophyll. Structure of chloroplast. Like mitochondria, chloroplasts are also double membrane bound membrane bound organelle having outer and inner membrane. The inner membrane is relatively less permeable to the substances than outer membrane and thus has more proteins including carrier proteins. The space enclosed by the inner membrane of the chloroplast is called stroma. Stroma contains a large number of organized flattened membranous sacs called thylakoids, which are arranged in sacs like piles of coins called grana or intergranal thylakoid. The thylakoids of different grana are connected by the flat membranous tubules called stroma lamellae. The membrane of the thylakoids encloses a space called a lumen. The stroma of chloroplast contain enzymes required for the synthesis of carbohydrates and proteins. It also contains small double-stranded circular DNA molecules and ribosomes. The ribosomes of chloroplast 70S are smaller than the cytoplasmic ribosomes 80S. Functions. The chloroplasts perform various functions like photosynthesis and storage of starch. Ribosomes. Ribosomes were discovered by George Palade in 1953 in the animal cell. They were observed under electron microscope as dense particles present in the cytoplasm. They are composed of ribosomal RNA and proteins and are not surrounded by any membrane. Structure of ribosomes. The ribosomes are composed of two subunits. One ribosomal subunit is larger in size and has a dome-like shape, while the other ribosomal unit is smaller in size and occurs above the larger subunit and forms a cap-like structure. The two ribosomal subunits remain united with each other due to the specific concentration of the magnesium ions. When the concentration of magnesium ions reduces below a critical level of both ribosomal subunits get separated. But by raising the concentration of magnesium ions in the matrix, the two ribosomes become associated with each other and is known as dimer. During protein synthesis, many ribosomes form a chain on a common messenger RNA and form the polyribosomes or polysome or ergosome. Ribosomes are of two types, 70S and 80S. Here, S. Swedberg unit represents the sedimentation coefficient, which indirectly measures the density and size of the ribosomes. The ribosomes of prokaryotes are 70S, while in eukaryotes the ribosomes are of 80S. In mitochondria and chloroplasts, ribosomes are of 70S. 70S ribosomes are made up of ribonucleoproteins in the ratio of 60 to 40% of RNA and protein. 70S ribosome consists of 30S smaller subunit, 21 protein molecules and 16S ribosomal RNA, 50S larger subunit, 34 protein molecules and 23S and 5S ribosomal RNA. 80S ribosomes are made up of ribonucleoproteins in the ratio of 40:60 of RNA and protein ratio. 80S ribosomes consist of 40S and 60S ribosomal subunits. 40S smaller subunit with 33 protein molecules and a single 18S ribosomal RNA. 16S larger subunit about 40 protein molecules and 3 types of rRNA. 28S, 5.S and 5S. 60S larger subunit about 40 protein molecules and 3 types of ribosomal RNAs. 28S, 5.8S and 5S. Functions. Ribosomes are the site of protein synthesis. The free ribosome synthesis non-secretory proteins, while endoplasmic reticulum bound ribosomes synthesis secretory proteins. Free ribosome synthesis structural and enzymatic proteins for the use inside the cell. The attached ribosome synthesis proteins for transport. Thus, these organelles are also known as protein factories. Newly synthesized proteins are processed with the help of chaperone proteins. Cytoskeleton. Cytoskeleton, it is made up of extremely minute, fibrous, filamentous and tubular proteinaceous structures which form the structural framework inside the cell. The cytoskeleton occurs only in eukaryotic cell and is involved in many functions such as mechanical support, motility, maintenance of the shape of the cell. Microtubules, microfilaments, intermediate filaments. Microtubules. The microtubules are found in the cytoplasmic matrix. They also occur in cilia, flagella, centrioles and basal bodies, mitotic apparatus, etc. Prokaryotic cells lack microtubules. The microtubules are hollow, unbranched cylinders generally about 25 nanometer in diameter with a hollow core of about 15 nanometer. The boundary of a microtubule is composed of 13 parallel protofilaments. Each protofilament is made up of alpha and beta subunits of tubulin protein, non-contractile protein. The assembly and disassembly of microtubules require GTP and calcium 2+. Functions. These help in the spindles and astral rays formation during cell division. These form the cytoskeleton of cilia and flagella. These help in generating the shape, rigidity and form of the cell and cell motility. Microtubules help in the anaphasic movement of chromosomes. Microtubules help in the intracellular transport of nutrients and inorganic ions. Position of future cell plate is determined by the microtubules. Microfilaments. The microfilaments are solid, unbranched, rod-like fibrils of indefinite length. They are mainly composed of globular protein actin, but have filamentous protein myosin also. They form an extensive network in the cytoplasm of cells and may be associated with plasma membrane. Functions. The microfilaments provide support to plasma membrane. These represent the contractile system of the cell and are involved in cytoplasmic streaming and amoeboid movements. Help in pseudopodia formation. These help in the formation of cleavage furrow during cell division. Intermediate filaments. Non-contractile hollow filaments of acidic proteins. These are also involved in formation of scaffolds for chromatin and in forming a basket around nucleus. Centrosome and centrioles. Centrosome is an organ usually containing two cylindrical structures called centrioles, which are surrounded by the cloud of amorphous pericentriolar material called centrosphere or kinoplasm. These two centrioles, called diplosome, are localized in the centrosome at right angles to each other. The centrioles are found in almost all eukaryotic cells such as animal cells, fungi and algae, but they are not found in higher plant cells. Structure of a centriole. A centriole possesses a whorl of 9 evenly spaced peripheral fibrils of tubulin. It is absent in the center, therefore the arrangement is called 9 plus 0. Each fibril is made up of 3 subfibers, therefore it is called triplet fiber consisting of subfibers A, B and C. The adjacent triplet fibers are connected by Ca proteinaceous linkers. The center of the centriole possesses a rod-shaped proteinaceous mass known as hub. From the hub, 9 proteinaceous strands are developed towards the peripheral triplet fibrils. These strands are called radial spokes. Due to presence of radial spokes and peripheral fibrils, the centriole gives a cartwheel appearance. There is no membrane boundary for a centriole, but it is surrounded by pericentriolar satellites, also known as muscle or microtubule generator or MTG. Functions of centrioles or centrosomes. Centrioles help in the formation of basal bodies which give rise to cilia and flagella. Centrioles form the spindle fibers that give rise to spindle apparatus during cell division. Cilia and flagella. Cilia and flagella are fine hair-like outgrowths present in outgrowths of the membrane. Cilia are similar structures which work like oars. The activity of cilia causes the movement of either the cell or the surrounding fluid. On the other hand, flagella are comparatively longer and responsible for cell movement. Flagella are found in both prokaryotic and eukaryotic cells but are structurally different. Eukaryotic cilia and flagella. Cilia and flagella are membrane-bound extensions of plasma membrane. They arise from the centriole-like basal bodies present at their bases like anchor and control their movements. They are cylindrical structures and their core is called axonium, which possesses a number of microtubules running parallel to the long axis. The axonium is present and made up of 9 microtubule doublets arranged radially along the periphery and a pair of microtubules present singly in the center. Such an arrangement of microtubules is referred as a 9 plus 2 organization. The pair of tubules present in the center are connected to each other by a bridge enclosed in common sheath called central sheath. The central sheath is connected to each peripheral doublet of microtubules by radial spoke. Thus, there are total 9 radial spokes. The peripheral doublets are also connected to each other via linkers made up of nexin protein. Now, I am going to discuss about the differences between cilia and flagella. The number of cilia per cell is usually very large. They are smaller in size. Cilia usually occur throughout or major part of the surface of the cell. Cilia helps in locomotion, feeding, circulation, etc. Flagella. The number of flagella per cell is usually 1 to 4. They are longer in size. Flagella are commonly found at one end of the cell. Flagella help in locomotion. Let's talk about the nucleus, the main part of the cell. Nucleus as a cell organelle was first described by Robert Brown as early as in 1831. Later, the material of the nucleus stained by the basic dyes was given the name chromatin by Fleming in 1879. Nucleus is a relatively large organelle controlling all the activities of eukaryotic cells. Some cells have more than one nucleus. Example, binucleate cells have 2 nuclei per cell, like paramecium, while multinucleate ones have many nuclei. Example, opalina. However, some cells lack nucleus at maturity, such as mammalian RBCs and sieve tube cells. Structure. A nucleus in non-dividing phase is called interphase nucleus, and a typical interphase nucleus is differentiated into nuclear envelope, nucleoplasm or nuclear matrix, nucleolus and chromatin. Nuclear envelope. It belongs to the nucleus on the outside and separates it from the cytoplasm. It is made up of 2 membranes, outer and inner. The inner membrane is smooth, whereas the outer membrane may be smooth, or its cytoplasmic surface may bear ribosomes like rough endoplasmic reticulum. The outer membrane is often connected to endoplasmic reticulum. These 2 membranes of the nuclear envelope are separated by a space known as perinuclear space. This space is about 10 to 50 nanometers in width. Nuclear envelope contains a large number of complex pores which are formed by the fusion of 2 membranes. The nuclear pores control the passage of substances to inside or outside of the nucleus. Example, RNA, ribosomes, proteins. Nucleoplasm. It is a transparent fluid and colloidal substance which fills the nucleus. It contains nucleolus and highly extended elaborate nucleoprotein fibers called chromatin. Nucleolus. It is a spherical structure found in nucleoplasm. It is not separated from rest of the nucleoplasm as it is not bounded by any membrane. It is the site of ribosomal RNA synthesis. Thus, nucleoli are larger and more numerous in cells that are actively involved in protein synthesis. Chromatin. The interface nucleus contains a loose, extended, and diffused network of nucleoprotein fibers called chromatin. These are named so because of their ability to get stained with certain basic dyes. Chromatin fibers condense to form chromosomes. Chromatin is essentially composed of DNA and basic proteins called histones. It also contains RNA and some non-histone proteins. The histone proteins are packaging proteins that are associated with packaging of DNA into compact structures called chromosomes. In higher organisms, the well-organized nucleus contains a definite number of chromosomes of definite size and shape. For example, a single human cell has approximately 2-meter long thread of DNA distributed along its 46 chromosomes. Structure of a chromosome. Chromatin. Structure of chromosome. A chromosome consists of two identical halves, the chromatid, which are held together at one point called centromere. The centromere appears as a narrow region called primary constriction of the chromosome. On the sides of chromosome, disc-shaped structures are present known as kinetochores. End of chromosome are called telomeres. They seal the ends of chromosomes and prevent their shortening or chromosome loss. Depending on the position of centromere, chromosomes can be classified into 4 types: metacentric chromosome, submetacentric chromosome, acrocentric chromosome, and telocentric chromosome. Metacentric chromosome. The centromere is present at the center, and thus during anaphase divides the chromosome into 2 equal arms. They appear as V-shaped. Submetacentric chromosome. The centromere is present slightly away from the center of the chromosome or nearer to the one end of the chromosome. As a result, chromosome is divided in one shorter and one longer arm. They appear L-shaped during anaphase. Acrocentric chromosome. The centromere is present very close to one end of the chromosome. Thus, it forms one extremely short and one extremely long arm. They appear J-shaped during anaphase. Telocentric chromosome. The centromere is present at the terminal end of the chromosome, and thus chromosome appears to have a single arm. They appear I-shaped during anaphase. A few chromosomes may have additional constrictions termed as non-staining secondary constrictions, or NOR, nucleolar organizer, near their ends. The part of the chromosome beyond secondary constriction is called satellite. A chromosome having satellite is called SAT chromosome, and these are considered as marker chromosome. In humans, 5 pairs of SAT chromosomes are present. Microbodies. This is the last part of the chapter. Many single membrane bound minute vesicles called microbodies that contain various enzymes are present in both plant and animal cells. They are associated with oxidation reactions other than those of respiration. They include peroxisomes, spirosomes, glyoxysomes. Thank you for listening.